Welcome to a walkthrough of Wiegand's dispersal tools for analyzing ecology data. Wiegand is a web-based platform for analyzing ecology data that requires no coding knowledge. In this step-by-step tutorial, we will show users how to use dispersal analysis tools to explore multivariate ecology data. Let's get started.
Click on the Dispersal module to explore multivariate data through dispersal direction coefficients, Beal's smoothing, and multivariate homogeneity of variance testing. Next, upload your dataset using the Choose File button. Alternatively, users can use sample datasets. For this example, we will select the dune sample dataset on grass species in a meadow. Selecting the dune dataset will also load the accompanying environmental data. Click Submit to proceed with your chosen data file. The Data Integrity Check page will confirm whether the data is usable, such as checking for correct sample and variable labeling. See our data uploading tutorial for more details on how to upload data and manage errors. Here we see the dataset has passed the integrity check. Scrolling down, the Data Overview page will also display a PCA plot and heat map of your data. Click the Heat Map tab to see a heat map overview, as seen here. The Data Editor tab can be used to make slight adjustments to the data. Here we can change the data type, edit the data, and remove entries. Find more details on our data uploading tutorial. Once ready to continue, scroll down and click Proceed. On the normalization page, you can select the normalization methods appropriate for your dataset.
There are 4 manual data transformation options to choose from. Click a radio button to select a methodology. Click Manual Normalization to apply your own data normalization configurations. A pop-up will show you variable data distribution plots comparing the effect of your selections. In this example, no normalization was applied. To make revisions to the normalization, click Go Back. Going back, we can alternatively choose automatic normalization to apply recommended normalization settings based on Pearson mode skewness. For example, here are the distribution results of the automatic normalization.
Click Export to save this plot. Each unique plot can be exported using the Export button. Select the export format from a drop-down menu. We can select from PNG, TIFF, PDF, SVG, and PostScript files. Next, select your desired export resolution. Options range from 72 dpi to 600 dpi. Finally, select export size of either full page or half page. Click Submit to generate the export file. The download link will now update. Click it to open the export file in a new tab.
To move on from the normalization step, click Proceed. For ease of replication, this tutorial will continue with no normalizations.
The second Data Integrity Check page verifies data quality in the supplementary data. This page is for our environmental data. Here, we see our supplementary data has passed the integrity check. Click Proceed to continue. After completing data pre-processing, you can select the type of dispersal analysis to perform. This example will start with dispersal direction coefficient analysis. We have now reached the main analysis page. Here, we can choose our desired plot and customize plot parameters. Select the coefficient corresponding to the desired distance metric, DD1 for Euclidean, DD2 for average, DD3 for chord, and DD4 for geodesic. The McNemar test is performed on presence-absence data to assess significant asymmetry between the two areas. Note that DD1 and DD2 are computed only for presence-absence data. Click Update to apply changes. Click on Customize Graph to modify the appearance of the plot. For this plot, we can first change the color palette. We can then change the position of the legend and its directionality. Click Update to apply your changes. Scroll down to view the customized DDC matrix heat map. Click Export to save your plot. Each unique plot should be exported using the Export button. Adjust the export parameters as before and click Submit to update the download link. Click on the Results banner to open a list of downloadable data files. Click on a file name to download. Continue to the Beals Smoothing submodule under Dispersal in the left-hand menu. Beals Smoothing calculates probability of viewing a target species at a specified site. Select whether to use original data and whether to include target species. Then select one target species. If no target is selected, all species will be used in the calculation. Finally, select a probability estimation type to specify if and how abundance values should be used in the calculation. Click Update to apply your changes. Click on Customize Graph to adjust the plot's presentation. The graphics center for this plot has some unique parameters. Select how to orient the plot. Select whether to rotate the X-tick labels. Adjust the color palette and legend as needed. Finally, click Update to apply your changes. Scroll down to view your Beals smoothing probability plot. Note that this example shows all species as a target species was not specified. Click Export to save your customized plot. Adjust the export parameters and click Submit to update the download link. Click on the Results banner to open a list of data files related to this submodule. Click to download. Finally, continue to the Multivariate Homogeneity submodule. The Dispersion Homogeneity box plot is for analyzing the consistency of group dispersions in multivariate data. First, select whether to use original or normalized data. Then, select whether to analyze presence-absence data or abundance data. Next, select a distance measure for calculating the dissimilarity matrix. Hover over the question mark for more information on each of the measurement options. Finally, select a grouping variable. Click Update to apply your changes. Continue to customize graph. Adjust the plot, colors, and legend as required. You also have the option of adjusting the number of rows in the legend. Click Update to apply your customizations. Scroll down to view the customized box plot. This plot illustrates site to centroid distance by groups based on a dissimilarity matrix. The x-axis represents individual levels within a particular group, and y-axis the site to centroid distance. Remember to export your customized plot. Click on any data files under the Results banner to download them. Finally, continue to the Dispersion Homogeneity PCOA tab. The Principal Coordinates Analysis PCOA plot is generated utilizing the same analysis methodology as the homogeneity box plot. The Principal Coordinates Analysis PCOA plot is generated utilizing the same analysis methodology as the homogeneity box plot. For this plot, there is also the option of adjusting plot accessories such as adding hulls, ellipse, spider lines, labels, and points. Click Update to apply your changes. Customize your graph as needed. In the Graphics Center, users can adjust the color palette and each of the plot accessories. Select the underlying statistics for the ellipse. Select whether to use a dashed or solid ellipse line. Next, choose whether to shade the hulls or leave them empty, and whether to use dashed or solid hull lines. For the spider arrows, we can alter their shape and change the line type. Finally, the centroid labels can either be enclosed or open, and the sample labels at the ends of the data lines can either be names or points. After adjusting these parameters, click Update to apply your customizations. Scroll down to view your customized Principal Coordinates Analysis plot. Click Export to download your customized plot. Click on the data files under the Results banner to download them. Access all data files from this analysis session through the Download option in the left-hand menu. Click Results to open a drop-down menu. A list of all downloadable data files from this analysis session will open. Click on any file to download. The download.zip file contains all listed files. Download the Wiegand Docker file to recreate the Wiegand coding environment. Download the RSource file to get the exact R script used during your analysis session. After exporting your results, click Exit to return to the home page. We hope you found this tutorial on the dispersal module helpful.